In this video, we'll talk about brain networks. Historically, neuroscientists have thought that cognitive brain function arises or comes from the idea that brain regions act independently. So a specific brain region would be associated with a specific function. There are limited examples of how this is true. For example, bodily movements can be pretty well localized, but now we're realizing that the overwhelming majority of cases, brain regions don't act alone. They actually work together in large-scale networks. We can define a large-scale network as structurally or functionally connected brain regions that are distributed across the cortex. So whether it be memory or emotion or decision-making, multiple areas are working together or in concert as networks, and cognition arises from the interactions both within and between these networks. Networks can be defined based on structure or function. Structural networks are based on anatomical linkage of neurons. So what are the actual physical connections in the brain? Another type are functional networks, and this refers to the activity in two or more regions where the brain activity are related or they co-vary together. So even when two regions may not have a direct structural connection, they can still be functionally connected. For instance, firing in the same types of patterns at the same time in response to the same type of stimuli. There are two terms that are pretty common when you come across brain networks. There are nodes and edges. Nodes are just individual brain regions. In graphs, they are visualized as dots. Edges are connections between two regions, and these are visualized in graphs as lines. So an edge is the connection between two nodes or brain regions, and networks are made up of many nodes and connected edges. There are different ways to define nodes. It's a little more scientific than just picking a random spot in the brain. An early way of doing this was to base nodes on an atlas of the brain, specifically the Brodmann atlas. The Brodmann atlas defined areas based on cytoarchitecture, or dividing up the brain based on cellular composition of brain tissue. This produced several regions of the brain. However, the regions are pretty large and it suffers from low resolution. Other areas break up the brain based on standard space, and a common one is to use brains in MNI space, and this stands for Montreal Neurological Institute, and they put out a standardized template averaged over many brains. They did this because in reality, everyone's brain is shaped just a little bit differently, and it's not exactly the same for everybody. So when creating brain networks, MRI scans are warped into this standard space, and we call that MNI space. Then neuroscientists can create networks that we know work for everybody in the same space. More recently, scientists have been using data-driven methods to parsilate the brain, and parsilation here is just another way of saying to break up the brain into discrete regions. So with data-driven methods, we can create everyone's individual networks. I just said that everyone's brain is shaped a little differently, so we can also capitalize on that and make a more personalized and precise network parcellation of your brain instead of a general network that could be a little misshapen from one person or the other. So it really depends on your goals. Do you want to make group inferences and use a standard space, or do you really want to get specific insights onto what happens at an individual basis and use these data-driven methods? So here's what some of the parsilations look like. Here are the Brodmann areas. The top two pictures show the lateral sides of the brain or the outsides, and the bottom two are the medial views or the middle of the brain. You can see all the divisions here, and we would treat every one of these numbered regions as nodes. Here's just another example of a parsilation. This one is using MNI space, and they parsilate these different regions, put a label onto them, and then again treat them as nodes. Now I want you to take a look at this parsilation. There are a lot more tiny divisions of the brain. So this is an example of a higher resolution parsilation. So not every parsilation scheme is the same. Some are low resolution like the Brodmann parsilation, and others like these are more high resolution. It just depends on the type of question you want to ask and how you go about answering it. Okay, so let's first touch on structural networks. With structural networks, the edges in these networks are long white matter tracks. The nodes are still brain regions, so these are just anatomically based networks. And there are a couple of ways that we can find and track these networks. The biggest one you should be aware of is through diffusion tensor imaging, or DTI. This basically measures the diffusion of water molecules running along major white matter tracks in the brain. And through this, we can visualize where these tracks are and what they are connected to. There will be a supplemental reading about what these major white matter tracks are, and you can find that on Canvas. Functional networks, as opposed to structural networks, look at brain activation or functional activation. So it's all about what's happening in the brain in response to stimuli, not just where the structural connections are. For example, say we put a participant in an MRI scanner and we have them look at a series of pictures, one after another. The brain activation as a result of viewing those images would be the functional activation you look at when constructing these networks. And edges here are a little bit different than with structural networks. Here in functional networks, edges represent functional connectivity, or the correlation between the activity in two or more nodes. So a functional network represents the connectivity or correlation between both distant and local brain regions in response to stimuli that represent our world.